Hello, everyone. My name is Dominique Maucieri, and I am a master's student in the Baum Lab at the University of Victoria. Today, I would like to talk to you about a portion of my thesis project revolving around how local and global stressors are affecting coral reef communities. Coral reef communities are very vibrant and diverse. The corals act as foundation species, forming the reef structure from their hard calcium carbonate skeleton on which other organisms are able to live. They are also ecosystem engineers, providing nutrients to their community in an otherwise nutrient-poor environment. As a coral reef community, there are many services that are provided to humans and other marine animals, such as ecological goods like fish and components of pharmaceuticals, the physical structure that the reef provides for organisms to live on, ecological services, such as being an important nursery, hunting ground, or breeding ground, and social and cultural services, such as tourism. Our reef communities are experiencing increasing effects from disturbances, and this is leading to the degradation of our reef communities. These disturbances can occur on a small scale due to local human populations, or they can occur on a large global scale due to the effects of climate change. However, these disturbances will very rarely occur on their own. Often there are multiple disturbances or stressors that act on an environment. There can be interactions occurring due to the effects of multiple stressors in an area, causing the resulting outcomes to often be unpredictable. There are many different ways that one can analyze communities to determine the effects of disturbances. One way is to examine the alpha diversity, or the number and abundances of species found in an area, and how that changes over a disturbance gradient. So for example, it would be the diversity in this site. And one could determine the alpha diversity of many sites and compare them. It is common in the literature to examine the effect of a disturbance by examining the changes in alpha diversity between sites, but commonly disturbances don't lead to a loss in species in an area, so it appears that disturbances have no effect on communities. However, when beta diversity is analyzed and the changes in species are taken into account, there can be effects of different disturbance gradients on the communities being examined. Beta diversity does not examine the number of species in a site, but how the identity of species changes between sites. Beta diversity therefore quantifies differences in the species composition between sites and can provide insights into whether stressors lead to homogenization of sites in which sites become similar to each other, or the opposite, biotic heterogenization, where sites become more different from each other by having less shared species and more unique species at each site. This latter phenomenon may suggest that stressors do not have the same effect across all sites. In corals, large global stressors can lead to biotic homogenization, where the species identities become similar between sites, as stress-tolerant corals are the only species to survive. The contrast is known to occur in coral microbiomes, where increases in disturbances can lead to increased heterogenization or less shared microbe species between samples, and more unique species. Little is known about beta diversity in coral reefs during disturbances, so by examining the beta diversity of coral reef communities, it can help researchers to construct a better picture of what is occurring during a disturbance. It is important to understand how disturbances are affecting reefs, because if we have a better understanding of the effects that disturbances are having on corals, we can implement more effective conservation and restoration actions. This fueled my research and led me to examine how local and global stressors affect the beta diversity of coral communities. I hypothesized that the local human disturbances and global human disturbances would lead to homogenization of reef communities. And again, this is due to the disturbance causing less stress-tolerant coral species to die, leaving more stress-tolerant coral species, which would be common between sites, making it so that there are more similarities in coral diversity between sites and lower beta diversity. My second hypothesis was that I would see an interaction between the local and global disturbances, such that the effect of heat stress on coral communities depends on the level of background disturbance the corals were exposed to. And this is due to there being multiple stressors occurring simultaneously in my coral communities. My study site is Christmas Island, located just north of the equator and about 2,000 kilometers south of Hawaii in the Central Pacific Ocean. My lab has been surveying the coral communities on this atoll since 2007 with 14 different expeditions. For my analysis, I analyzed the coral communities on a spatial and temporal scale in order to examine the local and global disturbances that had occurred on this atoll. There is a natural spatial gradient of local human disturbance, as nearly the whole human population is concentrated on the northwest end of the atoll. The local human disturbance has been categorized into 5 levels of human disturbance, shown as the different colors on the map of the atoll. There are 40 different sites, although all sites were not sampled in each year due to time and weather constraints. After several years of studying the benthic coral communities on Christmas, the atoll was hit with one of the most extreme heat waves on record, the 2015 to 2016 El Nino. This map shows the accumulated heat stress during 2015 to 2016, with the darker red meaning more heat stress, and Christmas Island is located in the center at the white triangle. This heat stress event led to approximately 90% coral mortality and transformed the reefs around Christmas. Although devastating, this heat stress was overlaid on the pre-existing gradient of local human disturbance and provided a natural experiment with which to test how these different stressors interact to drive coral beta diversity. I've been analyzing data from 8 sampling periods before, 3 during, and 4 after the heat stress event, allowing for a good baseline of coral community structure to be established and the effects of the event to be analyzed in detail. My lab has been collecting photos of benthic coral quadrats since 2007. They've taken around 15 to 35 quadrat photos at each site. Since different size quadrats were used in different years, all were cropped to 0.54 meters squared and approximately 54 randomly generated points were put onto the images, maintaining a density of 100 points per square meter. I worked closely with the other researchers to manually annotate each point using a program called CoralNet. Measures were taken to ensure all species identifications maintained consistency. To assess the effects of local and global disturbances, I performed 2 different analyses. First, a distance-based redundancy analysis with a permanova, which will help to visualize similarities and differences in the coral community structure at different sites and in different years. And the second, a temporal beta diversity index, which will allow the breakdown of beta diversity into species gains and losses. I used a distance-based redundancy analysis to analyze changes in community composition on both a temporal and spatial scale in order to investigate the local and global stressors in my coral communities. A distance-based redundancy analysis is a multivariate analysis that uses dissimilarity indexes such as the Bray-Curtis and Jacquard dissimilarity indexes to group sites in my data set together based on similar community structures. So more alike site communities will be grouped closer together and less alike site communities will be further apart. And I paired this analysis with a permanova, which is a multivariate statistical test, which allowed me to test if different explanatory variables were significant in essentially the clustering of site communities. Looking at these graphs, there are 2 different types of data. The left is abundance data, so the proportions of each species of coral were analyzed, while the right plot is occurrence data, so only analyzing whether a species was present or absent at a site. On these plots, you can see the before years, shown in red, are clustered closely together with the first sampling period during the heat stress event in 2015, shown in green. Then the other 2 green sampling time points during the heat stress event clustered closely with the time points from after the heat stress event, shown in blue. The red line connects the sites in chronological order from 2007 to 2019. The effect of year on both abundance and occurrence data for community structure was significant, showing that the community composition differed due to the heat stress event. Then, using that same redundancy analysis, I also examined the individual sites for each sampling year, which I have color coordinated here based on disturbance category. The red sites are the very high local disturbance sites, which group closely with the blue local disturbance arrow, especially in the abundance data, showing that the very high local disturbance levels affected their site compositions. The effect of the local disturbance was significant, and the redundancy analysis showed that in the abundance data, there was little change in the clustering of the very high local disturbance sites when subjected to heat stress, whereas corals in the other disturbance categories underwent large changes in community composition due to the global heat stress event. This effect was less pronounced in the occurrence or the presence-absence data shown on the right, suggesting the abundance of species greatly differed before, during, and after the global heat stress event. There was also a significant interaction between the local human disturbance and the global heat stress event, which, due to the low amount of change occurring at the very high sites, is likely an antagonistic effect, meaning the combined effects are much less than you would expect by adding the effects together. The redundancy analysis showed that there were differences in the community composition due to the effect of the global heat stress event, as well as the local human disturbance. Also, based on the differences in the changes before and after the global heat stress event in the different human disturbance categories, there is an interaction between the disturbances, which is likely antagonistic or producing smaller than expected effects in some sites. The second analysis I did to investigate my hypothesis that the coral communities would homogenize due to the local and global disturbances was the temporal beta diversity index. This analysis allowed me to break apart the beta diversity into losses and gains in species over time, as it uses a pairwise beta diversity comparison of each site community composition to its composition during the baseline year, 2007. Since it is comparing on a site level, there needed to be consistent sampling of sites in every sampling period. This is why there are 5 sites used in this analysis, and within those 5 sites, there are very low, medium, and very high disturbance categories included. This method will allow me to examine any changes in beta diversity over time compared to the 2007 baseline and examine the effects of the global heat stress event, which is the years in the gray rectangle on the graphs. There is again both abundance and occurrence or presence absence data being analyzed, with abundance on the top and occurrence on the bottom. Both graphs show that there was an overall increase in beta diversity between sites after the heat stress event, shown by the increase in the black line during and after the shaded gray box. There was also an increase in species losses due to the heat stress event, shown by the increase in the blue line over time. And finally, there was a decrease in the species gained due to the heat stress event, shown by the drop in the red line during the global heat stress event. Then I split this data up into the very high and very low disturbance categories to see if patterns are the same in different levels of local human disturbance. First, in the occurrence plots, the very high local disturbance is on the top and the very low is on the bottom. These plots show the same patterns that were shown in the previous plots, with increases in total beta diversity and losses due to the global heat stress event, as well as a reduction in species gains. Comparing the occurrence data with the abundance data, again, the very high local disturbance sites are on the top and the very low local disturbance sites are on the bottom. They are similar, except that there is a higher amount of species loss and a higher beta diversity before the heat stress event in the very high disturbance sites. This is shown in the top left plot, where the blue line is above the red and there is no crossing of the lines like in the other plots shown here. This suggests that there was initially higher species declines and more year-to-year variation in the very high sites before the heat stress event occurred. To bring everything together for you, the analyses I have shared with you today show that on Christmas Island, the effect of the global heat stress event and the local human disturbance increased heterogeneity, which contradicts my hypothesis that there would be an increased homogenization or similarity in reef communities. This suggests that there are other site-level factors that are influencing how species are responding to the different disturbances, leading to altered community composition. This may have future influences on the recovery of reefs due to the different community compositions. The effects of these two stressors do not seem to be occurring independently of each other, but they seem to be interacting with each other antagonistically, making it so that the effect of the heat stress on coral communities depends on the level of background local human disturbance that the corals were exposed to. The effects of these two stressors do not seem to be occurring independently of each other, but they seem to be interacting with each other antagonistically, making it so that the effect of the heat stress on coral communities depended on the level of background local disturbance that the corals were exposed to. Now, I'm not done analyzing this data yet, and in the future, I'll be continuing to investigate that increase in heterogeneity and exactly what it means for the compositions of corals in each community. I also will be taking a look into individual species and how their abundance and occurrence differs over the spatial and temporal scales. Finally, I will look into the interaction between global and local stressors to see if there are corals that seem to be co-tolerant or co-susceptible to both disturbances. I hope this research will help to better understand how our coral reefs are being affected by various disturbances so we can protect and recover these amazing environments. I want to thank my supervisor, Dr. Julia Baum, and collaborator, Dr. Sam Starko. Their guidance helped me to piece apart the analysis I showed you today and many others. I also want to thank my lab mates in the Baum lab and the Juana's lab for their support, as well as the Christmas Island field team, who without, I would not have any data. Finally, our many sponsors who have made this and other research possible over the years. Thank you.